All right, hello to our friends joining us via recording. We are talking today about something called the Central Dogma. Oops, give me just a moment. The Central Dogma sounds a little bit funky. It sounds more complex than it actually is. The Central Dogma, in short, is just the way that your cells take your genetic information and turn it into proteins. We know from studying last week's material and from the quiz that we took that proteins are really important for a cell. They do a lot of important things. So today we're going to talk about the way that we can get that sequence of amino acids, the way that we can get our protein's primary structure determined. So let's start with this. Let me get my pointer for you. The Central Dogma tells us you have genetic information stored in DNA. We need that DNA to turn into what are called polypeptides, or in other words, what we talked about them as is proteins. Polypeptides means a group of amino acids that are connected to each other with a special kind of chemical bond. What was the name of that special kind of chemical bond that we see inside our proteins? It was the special kind of chemical bond that gave us a protein's primary structure. What's that special protein bond? Exactly. Yeah, the kind of bond I use to hold together amino acids inside a protein are called peptide bonds. So when this picture shows us these things named polypeptides, we're just saying, oh, it's a lot of things connected with peptide bonds. For me to be able to go from my genetic information stored as DNA into my proteins, my polypeptides, I have to build something in the middle that's called mRNA. So when I use this fancy term, the Central Dogma, what I'm talking about is going from the way we store your genetic information into a copy of that information into a protein that we're building. That's big picture what we're looking at today. To start, we need to discuss the kind of genetic information that's stored inside your cells. So we talked about in lab several weeks ago, the part of your cell where we store your DNA, where we store your genetic information. Do we remember where in the cell is the genetic information stored? Where do we find your DNA? Exactly. Yep. So the DNA that's inside all of your cells is found inside that large central organelle called the nucleus. When we have our DNA in the nucleus, it's in this structure called a double helix. So double helix means that I have two strands of genetic information. They wrap around each other, making kind of this silly pattern that we see here, the double helix. Hey, I want to show you. Let me take this off real fast to show you. You probably can't see because my view is really small, but today Dr. Allis is wearing her double helix necklace just for you all. So my double helix DNA necklace is what I got on right now. Two strands of DNA that twist around each other. This makes it really stable. DNA lasts a really long time. When we talk about the double helix of DNA with these two strands of DNA, here's an important word for us to know. I'm going to put it in the chat. The two strands of DNA, I need everyone to write down, underline, highlight, star. These two strands of DNA are complementary. Complementary. Saying that they are complementary means that they are not mirror images of each other, not identical. They're the opposite of each other. So up here in my strand, my blue strand up here has the amino acid A, adenine. In its neighboring DNA strand, we have a green nucleotide. We have thymine. We have T. In the blue DNA strand, we had a G here in red, which means in the red DNA strand, we've got a C, a cytosine. My DNA double helix is not identical. Each side is not the same. They are complementary though. So if you know that there's an A in this strand, we know that the other strand will always have a T. Or we know if this strand has a G in it, the other strand will always have a C. You can use the sequence of one DNA strand to predict the sequence of the other, but they are never, ever exactly the same. Complementary means we're not the same, but we can be used to predict the other one. Now, if you notice on my picture here, and you have this in your notes, we've got a whole lot of chemistry going on over here. I promise you 100%, I am not going to give you a picture of this and ask you, which nucleotide base are we looking at here? Or which nucleotide base are we looking at here? I'm not going to ask you that. So don't worry, you don't have to memorize it. Why I'm including this for you is to explain to you why every time we have an A in one side of my double helix, we will always have a T in the other side. Or every time we have a C in one side of the double helix, we always have a G in the other side. It comes down to the structure of these nucleotides. So notice on my base adenine over here, A, I have two places where I form hydrogen bonds. Remember, hydrogen bonds form between dipoles. So right here, I've got a positive dipole, and down here, I've got a negative dipole. I have two dipoles that are sticking out on my nucleotide. Well, over here on thymine, I also have two dipoles that are sticking out, a negative up here and a positive up here. If I have two dipoles on thymine and I have two dipoles on adenine, these dipoles want to interact with each other. We will always pair A with T and T with A because they both want to make two bonds. But compare that down here when we're looking at cytosine on the right and guanine on the left. Notice that cytosine and guanine actually make 1, 2, and 3 chemical bonds, or hydrogen bonds, excuse me. When I make 3 bonds, I either am a cytosine or a guanine. I always want to pair with something that has the same number of bonds as me. So when we talk about the double helix being complementary, when I ask you, hey, who pairs with adenine? The reason you can never tell me guanine or cytosine is because guanine and cytosine want to make 3 hydrogen bonds. Thymine only wants to make 2, which makes it perfect because that's what adenine wants to make as well. Hey, by the way, Dr. Aulis has a favorite nucleotide. Dr. Aulis's favorite nucleotide is adenine. I keep referencing it here. The reason that adenine is my favorite, don't judge me. I literally have a cat named Adenine. We call her Addie. So my Addie cat is an orange tabby. We'll talk about later when we talk about that molecule ATP. The A in it means that it actually has an adenine inside of it. So the energy molecule in your cell has this same chemical component as your DNA has, this adenine. So my spastic yellow tabby is accurately named. So she's only adenine when she's like being really bad, you know, like when she's on the table licking my plate. Adenine, get off the table. Otherwise, she's just Addie. So probably more than you wanted to know about Dr. Aulis, but now you know. All right. So let me take a moment, and I'm going to pause because I do want to make sure that you can do this. I'm going to give you about 30 seconds. You have this sequence in your lecture notes, or if you don't have the lecture notes printed, grab a scrap of paper really fast. I want you to try to build complementary DNA. Before I set you loose on that, remind me in the chat, if the DNA is complementary, it is or is not an identical match. If we're complementary, are we exactly the same? Yeah, we are not exactly the same, right? But I can use the sequence of one to predict the sequence of the other. So I've given you one sequence of DNA. I want you to work now on our second sequence of DNA down here. So let me pause. I'll turn off my camera so I'm not staring at you all weird. Give you about 30 seconds. Let's make sure we can build a complementary DNA strand here. If you have built that strand, can you send me a thumbs up or a smiley face? If you're still working, keep working. 30 seconds goes fast. I pulled out a watch. It goes fast. All right, so a lot of us, maybe not all of us, but a lot of us have written that sequence. Let me take some time with you here together. We'll do a little bit of this sequence. I'm not going to go through all of it, but we'll go through some of it here. So remember, when we are building complementary DNA, that we've got two sets of bases that always go together. So we've got Dr. Hollis's favorite base, adenine, right? Whenever I have an adenine, what do I always pair an A with? What does A always go with, since we're complementary? Exactly. A is always going to go with T, which also means, by the way, that the opposite's true too. So T is always going to go with A. These guys always go together. Which leaves my other two bases. I've got guanine, that's a G. What does G always pair with? Guanine always goes with, exactly, yep, guanine is always going to go with cytosine, going to go with C. And again, the opposite is true too. So let's go down to our sequence then. We start with a T, which means I'm going to put an A in my complementary sequence. Then we've got an A, which means I'm going to put in a T. Then we've got a C, so I put in a G, I put in a C. So you get the idea. We just carry on in that pattern. As long as you know this stuff right here in my little box, as long as you know that A and T always go together and G and C always go together, you're in good shape for building complementary DNA. So we went from one half of the double helix into two halves of the double helix. We have both sides now because we started to make that sequence. And remember, because they're complementary, if I know the sequence of one, I can predict the sequence of the other. Now, when we talk about genetic information, what we've talked about so far and what we just built together was deoxyribonucleic acid, or DNA. The other kind of genetic information that's involved in the Central Dogma is called ribonucleic acid. Ribonucleic acid is RNA. So when I'm comparing DNA over here on the left to RNA on the right, there's a few important differences. One of the important differences we can notice right away looking at them. When we talk about DNA, DNA always has that double helix. It has two strands. So we talked about those two strands that we see here. When we talk about RNA, RNA only has one strand. It's what we call single-stranded. So one difference between them, DNA is double-stranded, RNA is single-stranded. Another difference between them is the kind of sugar that we use. So this part here where we see the colors, and same deal here where we see the colors on our nucleotides, that part is the bases. That's where the letters come from. But the line that we see here in beige and the lines that we see here in red and blue have sugar inside of them. And the kind of sugar they have inside of them is different. That's how they got their two different names. So let me type in the chat for you here. When we talk about DNA, DNA has deoxyribose sugar. Deoxyribose sugar. So you can see most of that here in its name. Deoxyribose. That's the name of the sugar that's found here in the backbone. When we're talking about ribonucleic acid, the kind of sugar that's in that backbone is ribose. The only difference between deoxyribose and ribose is that deoxyribose has one less oxygen. That's how it got its name, deoxyribose. That is totally cool with most of the nucleotides. So most of my letters that I have in DNA, I also have in RNA. But there is one of those letters that I like to say is too cool for ribose sugar. Like it won't deal with ribose sugar. Does anyone happen to remember from their studying ahead, which of these four bases that we see right here do we not have when we're in RNA? Who's not in RNA? Yes, several of us remember from reading ahead, we do not have thymine. When we're talking about RNA, RNA has adenine, it has cytosine, it has guanine, but it does not have thymine. Thymine is like, I'm too cool for this. This ribose sugar doesn't work for me. So somebody that looks almost identical to thymine, not quite, but very close. We have a new base, just like Amush put in the chat for us here. We have a new base called uracil. So U for uracil. Instead of using thymine, we put in uracil. We put in a U when we're building RNA. Here's a little wink, wink, nudge, nudge from Dr. Ollis. If you ever see a genetic information sequence that has a U in it, if you ever see uracil, that always means that we're looking at RNA. DNA doesn't have uracils in it. And if we ever see a thymine, we know we're looking at DNA because RNA will never have thymine. So DNA and RNA, the easiest way to tell the difference, do I have U's or do I have T's? That will help me to know which one I'm looking at. When we talk about RNA, which is the one that has just single-stranded, it's the one that has the uracils, technically there are three types of RNA. So the first type is what we call messenger RNA, and that's really what we're going to focus on in our class today, messenger RNA or mRNA. So messenger RNA has those sequences up here, the bases here on the top, with the ribose backbone that we see down here. This is the kind of RNA that will take the directions from inside the nucleus to outside into the cell. mRNA transports information. We also have some RNA inside our ribosomes. So it's called ribosomal RNA, rRNA. We aren't really going to talk about this much, except to say that it's there. We will talk some, though, on Wednesday about this one over here, tRNA. Technically, its name means transfer RNA, but you might want to think about it as translator RNA. And that'll make more sense in class on Wednesday when we talk about the process of translation. But translation is when we take information from its RNA form and we turn it into a protein. That's the process of translation. When we're talking about, I'm going to back up real fast. So whiplash, we're going to go through a bunch of slides. Right here. Oops. When we're talking about taking our DNA and turning that DNA into RNA, we are doing a process called transcription. So first today, we're going to talk about this process called transcription. The purpose of transcription is to get the genetic information out of the nucleus and into the cytosol. Because for us to be able to turn that into a protein, it needs to go to the ribosomes, and the ribosomes live in the cytosol. So when I want to do transcription, I have to get directions from DNA into RNA because RNA can leave the nucleus. DNA is stuck in the nucleus. I cannot build a protein directly from RNA because it can't get out to where the protein builders are. So transcription is a step that I need to make sure that my genetic information leaves the nucleus and goes out to where I'm going to use it. So that's the framework here as we transition into talking about transcription. Our goal is to get that genetic information from a form that's stuck inside the nucleus, from DNA in the nucleus, into RNA that's in the cytosol. All right, here comes some more whiplash going real fast. To right here. Your genetic information inside your cells is stored inside things called genes. So genes are specific chunks of information. We've got genes for hair color, for eye color, for skin color. We've got genes that control how tall you are or how short you are. All of those things are dictated by little chunks of information called genes. Let's say we're inside one of your cells. We'll say it's a skin cell. So this particular skin cell is needing to make that keratin protein, the protein that makes them so strong. So we've got a gene for keratin right here. And this cell says, hey, I need to build some keratin. I need to build this protein. To help your cell know where it's stored that information for keratin, that gene for keratin, we have these things called promoter regions. And when we think about promoter regions, I want you to consider those kind of like those billboards that you see on the side of the highway. Now, Dr. Ollis is going to age herself right here when I ask this question. Do I have any other friends here in class today who were driving before they had a smartphone? Was that anybody besides Dr. Ollis? I'm one of those old ones. Okay, I got a few friends. Awesome. Okay, so for my friends who used to do driving without a smartphone, I now rely on mine. We know the power of billboards, especially in the past. So you all know that I'm from Oklahoma most recently. I did a lot of driving between Oklahoma and Texas to visit family. So driving between Oklahoma and Texas, for me, when I'm doing that drive, there's something about a mint chocolate chip shake from Brahms that just does it for me. Like that is my road trip food right there. So if I'm doing a drive and I'm going from Texas to Oklahoma or Oklahoma to Texas, I am looking for the billboard that tells me where I'm going to find Brahms, where I'm going to get that mint chocolate chip shake. When I see that billboard, it says exit here for delight and happiness. Actually, it just says Brahms, right? But still, I know to get off the highway to go get what I want. That's the way that a promoter region works for genes in your body. It helps your cell know, where do I store that information? So what do we think? You all might not be Brahms people. What is a billboard that you all, if you saw it on the side of the road, would always make you pull over? Where do we like to go when we're road tripping, if it's not Brahms? Okay, Danae is a Brahms person. Like it. McDonald's. Hey, I got kids. I definitely do McDonald's. Buc-ee's. Yes, yeah. Oh yeah, Adriana's mentioning the fried pies place. Mm-hmm. Yeah, so we got a lot of Buc-ee's love in here. Love it. So next time when you're driving on the highway and you see a billboard for somewhere that makes you want to stop, you've just seen a promoter region. It's a thing that tells you where you need to go to get what you want or to get the gene that you're looking for. Excuse me. So here's how it works. I've got my gene right here. I've got the information that I want or the food that I want. Yeah, Seth is mentioning the beef jerky guy. Yep. So the food that I want, the directions that I want for my protein are right here in the gene. I put that promoter region right here. I have an enzyme in my cell that I want you to underline, highlight, star, called RNA polymerase. RNA polymerase is a protein whose name tells me exactly what it does. Polymerase means builder. I'm building something. Hey, this is not a trick question. Help me out in the chat. What do we think that RNA polymerase builds? Based on its name, what might RNA polymerase build for us? Exactly. Several of us are thinking like, this question seems too easy to be true. No, it's true. RNA polymerase is the enzyme I use in my cell to build RNA, to build the information that can leave the nucleus. So RNA polymerase looks for the promoter region. When it finds the promoter region, it attaches to the DNA and it causes the double helix that we see here. It causes the double helix to open up because RNA polymerase is going to read the DNA. It's going to read the information that's there and use that as the blueprint for the RNA it's going to build. So step number one, RNA polymerase has to find the promoter region. Step number two, it reads the DNA that's found there, and that's what we see right here. Now, I specifically mentioned that because here's a really important idea. Oklahoma, Texas, we're kind of part of the tornado alley, right? So thankfully, as long as I've lived in Texas, we haven't had too many tornadoes. We've had a few. We had more in Oklahoma. If I'm doing my drive between Texas and Oklahoma, I don't have a smartphone yet. Tornado comes through, rips out the billboard. The RNA polymer, so the promoter region is gone on my gene. If I'm driving and I don't see that sign for Brahms, if I don't know that Brahms is there, I'm going to miss it. And it's the same way with the promoter regions inside your cells. If the promoter region for your gene for keratin, for the protein for your skin, if that somehow loses its promoter region, like there's a mutation in that region and now it doesn't have the billboard anymore, we're not going to express that gene anymore because we're not going to be able to find it. So I need my promoter region, I need my billboard so that RNA polymerase can attach to it and it can go through and start reading the genetic information. Now notice as my RNA polymerase enzyme is going through and reading the genetic information, it's building that RNA that we talked about. It's building something that can leave the nucleus. So RNA is built based on the sequence of the DNA. And it's built just the same way that we built the complementary DNA, except instead of putting in any T's, remember that we said that in RNA we use U instead. So if this was my DNA sequence that I started with, this is the RNA sequence that I'm going to build. I had a T down here, so I can still use A. A is totally fine. I have an A down here in the DNA. Normally, if I was building more DNA, this would be a T, but now I'm putting in a U. C still goes with G. G still goes with C. So the only one that's different is we don't put in those thymines. We use the uracils instead. Let's do some practice. Again, I'm going to give us maybe 45 seconds this time. You've got that same DNA sequence that we had from before. That DNA sequence now, I want you to turn it into RNA. Now we're building complementary RNA. We're doing the process of transcription. So let me stop talking for a little while. Let's transcribe or do transcription on this DNA sequence to turn it into RNA. whenever you finish transcribing, shoot me an emoji in the chat. I'm going to give us plenty of time here to work through this stuff. Shelby is so done. I like it. Awesome. Okay, well, it looks like a lot of us are wrapping up. If you're still working, I'm going to do kind of like we did last time. We're going to set up our rules, what always means something. So anytime I have a T in my DNA that I'm turning into RNA, what do I put into my RNA strand if the DNA has a T? DNA has T. Oh, so let's reconsider here. Well, we're doing this one right here. So my DNA strand has a T in it, which means that when I build my RNA strand, when I'm building what's complementary to this, yeah, there we go. Now we're going on the right track. I would put in an A. If my DNA has a T, my RNA is going to get an A. Several of you, though, are correct. We're talking about what would happen in normal circumstances. If I was building DNA, when I have this A right here, DNA would have a T, right? But we're not building DNA anymore. We're building RNA. And thymine is too cool for ribose sugar. So when I have an A, I'm going to put a U into my RNA. So whenever I have a T in the DNA, A goes into RNA. When I have an A, sorry, my writing is so terrible up there. When I have an A, I'm going to put in a U. G and C are still playing nice with us because remember, those two are both still in RNA. So just like last time, we're still putting in a G here. We're still putting in a C here. C and G don't change at all. It's just when we're talking about the T's and the A's, because remember, I cannot put a T in my RNA. I have to always put a uracil. Any questions about building complementary DNA or complementary RNA? Or shoot me an emoji, a thumbs up, something that tells me if we're doing good. Shoot that my way too. Ooh, my computer freaked out. All right, we're looking pretty comfortable with this. Good. So I'm going to go ahead and move us on then. Let me get my pointer back. All right, here's what I'm going to do. So Dr. Aulis made the wrong group work form for today. This is what I'm going to have us do in our group work. Let me check beyond here. Okay, here's what we're going to do. First, I'm going to explain to you my library analogy that hopefully you've already seen by working through the notes. Then I'm going to split us up into groups and very quickly build you the group work form for today, not the group work form for Wednesday. So let's chat through this last example, and then we'll do that group work together. For those of us who were driving before smartphones, you will appreciate this analogy. But for any of us that have ever done a book report before the internet was around or before you were allowed to use the internet, that's what this analogy is for us. So we're going to use an analogy of Dr. Aulis writing a book report when I was in elementary school. you know, back in the age of dinosaurs when there was no internet. When I wanted to do a book report for school, I had to go to my local public library to find resources. I didn't have the internet that I could use. And perhaps the greatest resource of all back in the day, before the internet, was this big set of books right here called Encyclopedia Britannica. So Encyclopedia Britannica had pictures, it had texts, it had everything that you could need about everything that we knew at the time. So obviously it's expanded greatly, right? Since I know I'm a dinosaur, it's really true. So I go to the library. I am doing a book report on penguins. I do love me some penguins. So I go to the library. There's Encyclopedia Britannica for all the letters of the alphabet. It's all the information is all there. I only care about, for my book report, penguins. So I'm going to go pick up the book particularly that has the letter P. And then I'm going to scroll through all the pages that are in there until I find the section on penguins. Once I found that section, I think, sweet, I've got all this information. This is perfect. But Encyclopedia Britannica can't leave the library. It's one of those special reference books that no one's allowed to check out. It's too valuable. So if I want to take this information with me out of the library to my house where I'm going to do my book report, I've got to go find the copy machine. Now, to be fair, that is not quite the copy machines that I had. I was like, let's go big on this like dinosaur motif, right? So my copy machines were slightly better than that one in the library. But I would take my nickel over to the copy machine, put it in there, and I would copy the pages that I want, the pages that have the information I need. Now I can leave the library and I can go out into the world and do my book report. This is a lot like what we see going on in our cells. Your DNA, all of your genetic information, is inside these encyclopedias. Those encyclopedias must be protected. We don't want to get stuff spilled on them. We don't want dogs eating them. Just like in your DNA, we don't want things like free radicals or bacteria or viruses messing with your DNA. So your DNA is stored in the nucleus, just like these encyclopedias are stored inside the library. There's a lot of different information inside the encyclopedias. I mentioned in your guided lesson, I did love to research the possibility of aliens. I loved to learn more about mummies. I love to learn about tornadoes. There's all kinds of different things that I love to learn about. But if I'm doing a book report on penguins, I don't care about the information about aliens or mummies or tornadoes. I only care about the information about penguins. Just like when your cell wants to make the keratin protein, it only cares about the information for the keratin protein, not the information for everything else. So we have our gene, we have our information about penguins that we're interested in. To get that information out of the library, we need to copy it. Or to get that information out of your nucleus, we need to copy it with RNA polymerase into RNA, into a thing that would be able to leave the nucleus. So as you read through that analogy again, when you're at home, try to consider it in those terms. The goal of transcription is to get the information from your nucleus, where it's stored, into your cytosol, where I can actually use it. Let me check out my chat before I send us to groups here in just a moment. Yeah, so Seth is chiming in on other old things, old time stuff. Yeah, when we didn't have those fancy projectors in the classroom, right, where you can modify a PowerPoint during class, you had to bring your little transparencies with you to class to project those. Yes, the good old days, right? So Dusty asked a good question. Let me see if I can put that to the class. So when we talk about the copier, true or false? What do we think? Is that RNA polymerase in our analogy? What do we think? I presume, Dusty, that you're meaning to say copier, right? Or were you meaning the copies that it makes? Okay, so two separate questions. I sent you in the wrong direction. So when we talk about the copier itself, that would be RNA polymerase, the machine that makes the copies that can leave the nucleus. That's RNA polymerase. When we talk about those copies, the pieces of paper that can leave the library, what do we think those pieces of paper would represent in our cell? What is it that RNA polymerase builds? Because that's what the copier is doing. It's building us a copy. Yeah, exactly. Yep. So when we talk about the copies, the things that can leave the library or the things that can leave the nucleus, that's the mRNA. So the copies that we built is the messenger RNA. The copier is RNA polymerase, the thing that builds those copies. When we talk about the specific section on information that we're looking for, that's like the genes in your cell. And your encyclopedias with all the information, that's like all the DNA that you have in your cell. Any other questions about transcription? That's the process we talked about today, transcription. All right. Well, while my breakout rooms are being built, you have this activity somewhere in your notes packet. Can someone help me out? What page do you have this table in your notes packet? My chat went away. Okay, page 3. Excellent. Okay. So when I open up my classrooms, my breakout rooms for you, go to page 3. You can start talking about these things with your classmates. I am really fast going to build you your Google form where you'll give me your answers to this. But since this is something that's directly in your notes packet, you can start talking about this now. You know what the activity is. So again, page 3, where you have this table, I'm going to build you a Google form to put in your answers. Once I have that form built, I will send that to you as an announcement to all of my groups. When you put in your information and you're submitted, we are all done with our lecture class. So remember Monday Lab Friends. At about 9:25, today's lab quiz will open up. Take that lab quiz before you come to our class today at 9:40. Otherwise, Wednesday Lab Friends, remember that we're going to have a quiz, a lecture quiz, just like we did today, Wednesday morning. Take that quiz, then come see me at about 8:10 on Wednesday. So I'm going to open up those rooms. Again, start working on this, page 3. I will see some of you in lab in just a bit.